Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, or then I was a fact checker for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated to aim at the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked in bacteria, but I've also worked as a research integrity specialist. I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology. Actually, we're going to move to every other week. So yeah, we'll make a reminder at the end and on our Twitter as well. And today we're doing an overview. Actually, it's a little bit different. We typically just like look at what's been done in the previous week. But this week, we're actually looking at everything in 2021, top 10 cited non-COVID papers.
Yeah, boy, it is going to be a special show because we are going to be looking at the top 10 most cited articles in 2021 that don't mention COVID, because COVID is something that we talk about a lot, and so it's nice to take a full week to just put that out of our minds and then try and look at the things that we might have actually missed because we were focused on COVID in 2021. And yeah, you can make sure to message us at MicroTWJC if you read a paper that you want to suggest to us as well to cover in the show for future episodes.
You can follow along with any of the papers that we discuss in our shared Zotero library linked in the doobly-doo below.
Yeah, and we really want to hear from you, so please use comments or tweet us at MicroTWJC. Right, so let's get into it, because this is going to be a kind of different show from what we're usually doing, because what I did was I went into a scientific literature kind of database called Scopus. I always pronounce it weirdly. But yeah, and I looked for the top 10 papers that were not related to COVID. So doing searches for fungi, bacteria, viruses, and then like searching through the papers which had the most citations, weeding out ones that were like not original research, weeding out ones that were just not related to, and also like weeding. So we also, all the papers we're talking about here are open access, because if you can't read it, we won't read it. And yeah, and without further ado, I rank them in order of citations. Now, obviously, citations they will change during the rest of the year, because these articles will still keep gathering them. But for this moment, I'm looking at how it appears right now in the middle of 2022, when essentially gives all the articles a much more even chance of getting citations. Because if I did this in December, something published in December wouldn't get as good a chance. So this is not just going to be looking at the research, but also we can delve a little bit into like the impact this research is having, what the fact that these articles have been chosen, the reasons why these articles have gotten these citations, why are they important?
And it's interesting too from the standpoint of this is just a snapshot, and science occurs like when we're reading papers and adding things to a field, the fields could be like many decades old, and some papers that are like much older accrue citations right throughout their lifetime, but some were also maybe flashes in the pan. I think that that's kind of an interesting, like, seeing something just from the year's citations is really what got attention during that year. And I would say that as time goes on, I don't think necessarily these papers would still be the highest. I mean, some of them I do think, because of the way they're like catalogs. I think the catalogs will continue to accrue sort of citations for a long time. But some others may not, and there may be other papers that don't get caught in this list. I mean, as with any metric, right? It's difficult to capture very comprehensive understanding of an entire field, such a broad field, such as microbiology, to say the least. Any sort of understanding just from looking at the top 10. But yeah, I would think that there's probably stuff that is super interesting out in the literature that we just don't catch as well.
Yeah, which I think we can, without further ado, let's bring us to the first paper, which is titled "Cone-shaped HIV-1 capsids are transported through intact nuclear pores." And this one is at the bottom of our list. This one got 64 citations, was published in February 2021. So another thing you might notice as we go through this is that a lot of these articles are represented in the earlier aspects of the year. So that's something to keep in mind, because if something is like published in January, it's got more time to collect citations things in February. So it's kind of like almost a little asterisk. But even so, this is
Yes.
still, yeah.
But it's pretty cool. It's definitely something that we probably would have chosen as well if we had seen it come up just in our like table of contents searches. A lot is known about this process of HIV. So HIV is a very well studied virus, mainly because it had global pandemic status at some points, and it still is a current high burden of disease in a lot of different places. There's a lot of drugs that were created for this virus, and that's on the back of a lot of basic science research just to understand how the virus works in general. So it's a reverse transcription. It's a RNA virus, right? It's a it's an RNA genome that wants to go through a DNA intermediate.
It's a weird. It's an old duck of a virus because it has an RNA genome, but through its virus cycle, it turns itself into a DNA genome and integrates within the host nucleus. So it needs to somehow be able to get its RNA genome into the nucleus of kind of a bit of a big ask.
Yeah, it's super tricky because the nucleus has its own membrane that keeps things out. So in addition to having to fuse with the outside membrane of a cell, it also has to be able to get into the nuclear membrane. And so it's already known that the outer envelope, so it's an envelope virus, just like our favorite virus, SARS-CoV-2, our infamous new favorite virus. So it fuses to the cell. It loses a capsid. The capsid is a proteinaceous sort of. It's almost like a phage head, if that's like an analogy that you could think of for this capsid. And that's covered in proteins, and that migrates to the nucleus. And so that's known. It actually, it's one of those fun. Is this a migrating capsid?
Yeah. So essentially, what it does is this capsid hitches itself to a microtubule and goes towards the nucleus of the cell.
Yeah. So if people have seen those 3D animations, right, of like inside of a cell, there's all these motor proteins that grab cargo vesicles and move them along these tubules. This is the same. This is thought to be the same mechanism that is moving capsids, these viral capsids, towards the nucleus.
Yeah, and these capsids want to get into the nucleus, and because they contain the genome, they contain a reverse transcription enzyme and integrase that it wants to deliver into the nucleus. But there is a problem, because in order to get in the nucleus, it needs to get through these holes in the nucleus, which are shown in gray on this like diagram, and these holes are the nuclear pores, where things go in and out of. The problem is, in all previous studies, they found that nuclear pores are actually much smaller than the HIV capsid. So it was thought that it's impossible for the capsid to actually get into the nucleus.
Yeah, and then there's all these competing hypotheses as to then how do all the things get in there, right? Like, is there partial uncoating? Does it like sit at the pore, like inject the things? And the authors use our brand spanking new. I mean, not brand spanking new. I mean, we've talked about it a bunch now, but it is a relatively new technique in the field of microbiology of cryo-electron microscopy. So that's freezing stuff in such a way that everything's like a snapshot in water, so that no weird solvents or other cross-linkers to induce any artifacts. Everything's frozen in this water structure. Very thin slices are taken and imaged using electron microscopy. You can even zoom through the layers. You get like a tomograph style. I know, so they're not zooming. You can even tilt the camera a little bit to sort of look at the angle a little, and you get these 3D reconstructions of what's going on.
Yeah, and because of the way it's done, where it's actually literally freezing the cell in situ, this is very different from what was done before, where you'd have to take a protein in isolation and then use various like imaging ways to try and get an idea of what shape it is. Whereas here, it actually shows what it's like in the cell. So it is possible that a lot of the proteins that were found through previous methods, like X-ray crystallography, might not be 100% representative of what they would be like in an actual cell. We tend to think of proteins as like these. I mean, with the way we image them, it's hard for us to understand like how kind of wibbly wobbly proteins are in real life. Like, I mean, I remember back in like kind of when I was studying university, we basically had these like sticks and ball like things that we'd have to put together to like get an idea of what molecules were like.
I think people still use those like molecular models to get a sense. Yeah. But also that's like on the molecule. Proteins are like so many molecules
Yeah.
large. It's really hard to construct a protein like that just to get a sense. And again, those are that's our representation in plastic. We don't really know like the rigidity inside of a living system. And this data, cryo-EM, really allows us to capture that more. So they just look at it they do a bunch of different ways of looking at it, and they're just collecting tons of instances where they see things. So they look at like, I don't know how many hundreds of these slides, but they usually go down and they're looking for specific events. So like looking for capsid structures and looking for capsid structures near the nuclear pore. That's like what they're curious in looking at. I think they also even use they use a technique where they make a mutant that it's a mutant of the cell, isn't able to import things into the pore for some reason, and so they end up trapping a lot of the capsids just outside of the pore to get a sense of like where exactly in relation to that pore are these capsids. Essentially, enriching their sample for the event that they're curious about.
Yeah, and I think what they do find is that they find that there are capsids that end up, and
Yep.
So I think I'm gonna scroll down to see the actual 3D image of that.
Yeah,
figure
3, I think, has a great image.
Yeah, I'm currently on video S5,
video.
which you can kind of see it built up, where you can see that one of the virions has managed to get its way through the nuclear pore, which is something that people didn't think was possible, because it's cone shaped, so the back end of it is much bigger than and they think that that's partly because of the way it's built up, because the HIV capsid has these hexamers, which are like kind of these kind of like Lego bricks that make up the capsid. So part of it is made of these hexamers, and part of these made of these pentamers that are arranged in a certain way. So you get more of one towards the like kind of beginning of the cell capsid than the other.
Yeah. And I think even from these images, like the figure you're showing now, they're able to look at the electron density of these EM images and guess at, or be able to infer that in some of these capsids that are getting lodged in, there's like structural movement a little bit, like stuff's dissociating or kind of flexing a little as it hits the surface. And again, like basically they're trying to take this data and figure out what exactly is going on, because having the capsid just like seem to lose a bit of integrity at this point could be the hypothesis that it just dissociates like right at this point, or maybe one end of it dissociates, right, and everything flows out of one end. But I think they settle on the idea that it actually goes inside of the nucleus itself, the whole kit and caboodle.
Yeah, it's interesting. Inside the nucleus, they find like I think empty capsids or something akin to that, where there are capsids, but that are just completely empty.
Yep.
So somehow it comes that maybe like it's as it goes into nucleus, it like kind of there's a toothpaste effect where like it gets squeezed through. But
yeah,
I think what they is that what we thought the size of nuclear pores were isn't actually what we think, because I think those original nuclear pore measurements were based off of data where it was collected outside of a outside of a cell, and because of that, it doesn't replicate so in the actual cell, the pores are under a lot more tension because they're in a membrane, and so based on their measurements, they say that it's actually, I think, 4 to 9 nanometers larger than other measurements.
Yeah.
Would think it is.
Yeah, absolutely. And so maybe it's not such a crazy idea to think that the pore can just have the whole capsid slip right through. although it is clear from the data that something is happening to the capsid as it goes through that pore, which probably makes sense, right? There's going to be protein-protein interactions during that period.
Yeah, I think there is like some aspects. So I think the capsid, there's this part of the nucleus I think they call the nuclear basket, where which might interact with some of the capsid proteins on the front of it. So, I mean, I don't think it's an accident that all the imagery we've seen has the HIV virus like kind of
Oh,
oriented
yeah, with
like in a
the
sharp
narrow tip.
burst.
Yeah, there is a polarity to these. They don't go in any direction.
Yeah, so I do feel like that, and that seems to be like an important aspect of it.
Yeah.
Um,
But yeah,
so
that's all I really wanted to say about this. I think it's kind of fascinating to see that through studying these capsids going through the membrane, we end up learning a whole bunch about the membrane itself and the pore itself. And that's a theme that comes up in virology a lot because the life cycles of viruses are so closely matched to our cells. They're dependent on us in so many ways. And so you end up learning a lot about just regular cell biology by studying viruses doing their thing.
yeah, and I think it's quite easy to see why this is such a big discovery, because this is an aspect of the HIV life cycle that was very much like unknown or a mystery. And so this kind of being unveiled, I can imagine that lots of other researchers working on this might find this relevant
Yeah.
and think, oh, this interaction, could we drug to prevent this? Could we do other things to, and or even to build up on it? So I think that this has earned its place on the list, I think.
Well, I would hope they all earn their place in their own way.
Yeah, in their own way, but I think this is the most easiest to see why. For me, I think this is very easy to see why it's here.
Yeah, with our infectious disease sort of biases and
Yeah, true.
loving the field of understanding viruses or understanding these tiny little monsters, monsters.
Yeah. Monster means like unknown, I guess, or like kind of unknown. So yeah, I can think that makes sense.
Yeah. All right. Next up on the list is partial recovery of visual function in a blind patient after optogenetic therapy.
Yeah, 78 citations published in May 2021. So I think this one made a bit of a splash. And at first I was wondering, why was this? Because I was looking for microbiology papers and trying to understand why did this fall under the remit of microbiology. And then I kind of read into it and realized, oh, this is gene therapy,
Yes.
where they used an adeno-associated virus to deliver a gene from an algae into a person.
It does sound pretty crazy. And then using specialized goggles that specifically replicate the images that the goggles see into the wavelength of light that the algal protein is sensitive to, and projecting that onto your retina is what recapitulates sight essentially in this model.
Yeah. So what we have here is we have a patient who has retinitis pigmentosa, which is a neurodegenerative eye disease where loss of photoreceptors can lead to a complete loss of sight. And so this
So they really don't have the proteins or the cell type in their eye that detects light molecules. Like those cells are genetically defective and thus non-functional.
Yeah. So what they in this instance they're using an optogenetic kind of deal where they've got a protein that reacts to light from algae. So this protein that is called let me look this up quickly.
Crimson R.
Crimson R. So they originally came from a unicellular algae, which allowed the single cell organism to detect and move towards sunlight. And then it was engineered to kind of make it better. So it was engineered to like make it respond to red light, I think.
Yep.
And
then, strangely enough, they also fuse it to red fluorescent protein. And they say that that's because it helps for the expression for whatever reason.
Right. I mean, there could be a number of reasons. I mean, there could be even like, yeah, I think expression
It could be like a fret type thing. Oh, I was also thinking maybe it's also helpful. Maybe it helps modify the wavelength that the Crimson R is responsive to as well.
Yeah. Sometimes like, because I mean, that even happens with like plants where you have like accessory like fluorophores, which can broaden the kind of lights that
yeah, the sensitivity spectrum of those particular proteins. Yeah.
Yeah. So effectively, what these proteins can do is they can detect amber light. And
But the
and it,
world isn't made up of amber light,
yeah,
so the second part of this is goggles that just translate the sort of the lights and darks of the world into this amber light that they can shoot into the eye. Oh, there's a video?
yeah, there are
I
two
didn't see
videos.
this.
So they took videos of this gentleman wearing trying I think after he was received the treatment. So this was so this is the first one where he was given the treatment, which was an adeno-associated virus. So adenoviruses, well, adeno-associated viruses are different from normal viruses because they're part of the
Mm-hmm.
dependoparvovirus family, where effectively they're dependent on another virus to cause an infection. Um,
Mm-hmm.
so they're ideal for gene therapy. So what they've seen is that person, he couldn't see, but now he's got the goggles on. He has to be able to determine. And I think on the laptop, then you have a like a little monochromatic image of the, because these goggles are like kind of spraying. They're directing the image directly on this person's retina, and I think
Yes.
the laptop is showing what he's supposed to be seeing, and it's going to be a very black and white image, and also like has quite low resolution as well.
Mm-hmm.
And then you can see him like he's having to scan around because it kind of is only affects a small part.
I do want to say with the adeno associated, there's already, so like this, there's like a subset of this particular disease where you lose the pigments and that's why the photoreceptors aren't working. And so then there's a gene therapy already approved for that.
Mm.
The brand name is Luxturna. It's a adeno-associated virus that delivers a construct that allows those cells to make the pigments again. But this strategy is different than that because it's really almost recapitulating like the signal chain. It's not trying to make the cells recover in any way. Essentially, it's like replacing the sensing mechanism that those cells have with this artificial one that's amber light and photoreceptor and crimson R.
Yeah, which also like, I mean, one of the interesting things about this is because it's using an entirely new photoreceptor to react to light. There is almost a possibility of, well, what other photoreceptors can we add to people to make people sensitive to different wavelengths? Um,
Yeah, there's definitely an interest. I feel like there is an enhancement. So like sometimes when we talk about genetic therapies, like people can talk about like therapeutics, but then also enhancements. And like there's like ethical differences, right,
yeah,
between wanting something for enhancement versus wanting something for therapeutic purposes. I do think that this borders on that because yeah, like I guess in some ways if I don't know what it would be like if a healthy person got this particular gene therapy, but would you become sensitive to this other wavelength in some ways? I'm not sure. Yeah.
yeah. Uh, but I think the
But
reason why they did it was because they didn't want to integrate. So it can be quite difficult to correct a defective gene. You need to have it integrate in the right place. And in this case,
mhm.
they can try. They don't have to have that level of specificity to do a correction.
Yeah. And I mean, I think the other thing is they're targeting people in the late
Mm.
stages of their disease. So with the old, it's not the old, it's not even that old, with the currently approved methodology for this, first of all, it's only a subset of patients that will benefit from the gene therapy. And it's only those patients that haven't yet, like it was used mostly on early onset, like before it got bad or like on during its development. And so I think maybe this is also a way of trying to broaden, like it could work on many people because because of it's kind of independent from the cell development part. We're not trying to work with the biology here. We're sort of using something orthogonal to the biology in order to
Right.
replicate. Yeah.
Yeah. So again, these are like a number of tests that they're running on this individual, and again, we should have pre-emphasized this is just one person that's being
One
tested.
person. One person.
So yeah, one person.
And
But
yeah, and they go through a bunch of these very functional tests to be able to discriminate objects. And it seems like it has a huge effect, right? Where they can't see anything in the control sessions, either either like without, because like they only do it in one eye, I believe, is how it's done. So like one eye is the control eye, the other eye is the experiment eye, and they can do that with or without or they can use the goggles on both of those, and get a sense for where the effect is happening. They also measure that effect in the brain. They also attach electrodes, and they sort of do a lot of complicated signal processing to say, like, if this particular specific brain signal fires, then it must be working. Or it's what is considered like I guess the optical center or something like this. And only in the condition where both the eye is treated and the light is shooting onto the eye that's treated do you get that particular signal.
Yeah, the occipital lobe is like right at the back of the brain, so like it's quite. I mean, again, it's probably the most awkward, like in the way that most human biology is. It's in the most awkward place possible because you'd think, oh, the place that handles sight would be right near the eyes. Nope, it's on the other side of the head.
Yeah.
But yeah, they do measure that, and they do find a reaction to it. And again, this is like, I can see why this is, because I mean, making a blind person see is literally like almost like a biblical miracle.
Yeah, there's
so
like
it is
heavy sci-fi vibes, I feel like I'm getting off this paper. It's a very exciting and like sort of even to the layperson, like sounds very cutting edge, right, in terms of what's happening here.
Yeah, I mean, even to me, it sounds cutting edge, and
we are lay people in this field, I'm pretty sure.
Yeah, that's true. But yeah, even to us, this, I mean, they've essentially giving people an entirely new photo, like incredible.
But yeah, I guess like, you know, I feel like in terms of like where I think this technology, I'm like, there's like, thinking about the ethics of this type of technology, having it go into production. And this is just one person as well.
Yes.
Like, how does that actually work? How do these goggles, right? Like, how will the goggles be regulated? And how do you make sure the goggles are something that is easy for folks to use? It definitely has a long way to go, but the excitement around being able to do this at the technological level, I'm sure, is what's driving this paper's citations.
I agree completely. I feel like gene therapy is somewhat like coming of age at this point, where
it's very early. We're getting some of the early starting. Actually, the next paper actually talks about this. Are we ready to move on?
Yeah, I think we're ready to move on to the next paper. Which I'm gonna like, hold on, I need to turn this video off.
Okay, I can read off the title. It's a long-term study of AAV gene therapy in dogs with hemophilia A identifies clonal expansions of transduced liver cells. So yeah, so gene therapy is already here, by the way, right? As I mentioned in the last one, there's Luxturna, which is something that has FDA approval, and it's for a very specific type of eye defect. This is something that's gone through phase 1 clinical trials, I believe, and it's to have your cells produce factor 7, which is a clotting factor. So it's people with hemophilia. They need typically we give therapeutically a clotting factor. It was thought, why don't we have people just make their own clotting factor by transforming their cells? And so phase 1 clinical trials have been done on this. But of course, that's just like, the follow-up period, it hasn't been very long yet for those phase 1 trials. But in dogs and mice, presumably, where this was first developed, they do have long-term follow-up study information about this particular construct, this genetic therapy, and so they're doing a lot. This is the published paper that gives us some of those long-term effects. I think 10 years is how long these dogs have been around. 10 years with AAV that inserted factor 7 expression into their liver cells.
Yeah, it's really fascinating. So they did like I think there are 2 different ways they did it. They used a 2-chain strategy or a single-chain strategy, and they used AAV. So adeno-associated, it ejects its genome into the cells, and then the genome kind of like is supposed to stay outside of the cells. It kind of lives out in like the cytoplasm where it produces the protein. But or like a
plasmid.
Yeah, kind of it's meant to be like episomal, right? So
Yeah,
it's meant
episomal.
to like, so it's, so
That's
the
also
idea
how
is,
Epstein-Barr virus is a virus that likes to do this sort of thing.
yeah, and so the idea is they're concerned about long term because long term they're thinking that since cells are dividing, that episome might be lost during that time.
Mm-hmm.
Uh,
Mm-hmm.
and I think these initial tests in dogs were because in dogs hepatocytes the liver cells tend to not be as highly replicative. So the
Mm-hmm.
idea is like these dogs model be like well designed for understanding the durability of these uh, episomes. And I think
Yeah.
what they found was quite interesting. But I mean, firstly, the dogs didn't have any serious adverse event directly involving like genotoxicity. So the thing that we're worried about
Mm-hmm.
for gene therapy is if your gene therapy vector integrates in a random piece of genome and gives like your subject cancer or other sorts of things that kind of mess with how the genome works.
Yeah, all sorts of strange effects from either integrating in the wrong place or interacting in some way. Yeah. And I think there was some evidence in mice that that could be something that happens. And so it
Yeah.
is thought that in this long-term period, if you don't lose the episome, you may get it integrated somewhere. The question is whether or not those events are dangerous.
Mm.
Mhm.
Yeah, and I think that so at specific points during like I think they had regular tests of these dogs of their factor eight activity,
Mhm.
and they found that some
Oh.
dogs the factor eight activity actually like increased quite a lot, and all the dogs still tested positive for it. And I think in terms of like their health, they was doing better with this factor eight like production than without it. So I
Mhm.
think in
Mhm.
terms of like they call them bleeding events. I think there was a reduced there's a the amount of bleeding events these dogs experience throughout their lifetimes was much lower
Yeah.
than they would if they didn't have the therapy.
Yeah. Yeah. Oh, yeah. I misspoke. Factor 8. I was saying factor 7. Maybe Final Fantasy 7. Anyways.
There's lots of I's in there. They're written in Roman numerals, and so
But yeah. So yeah, it seems like the dogs are fine. They do find integration in some
yeah, I think
genes. Um,
that's the
yeah.
main thrust of this paper, where they look at the integration genes oath, and I think when the dogs have passed, they look into it and they find that there are lots of integrations in different places, just not in places that would cause. I mean, yeah, I think that
Yeah, not in places. I mean, actually, I think they do think that they might be places that cause
yeah,
problems, but they don't see any pathology. And the dog's liver test, like the health when you assess liver health in a dog, there isn't anything that is out of the ordinary besides 10 years of aging that's happened to these dogs.
yeah. But from the perspective of the integration sites, there are in some of the dogs there were like versions of the virus that were integrated in them, and the virus itself kind of
Hmm.
like during the integration process, its genome can get chewed up a bit. So I think that they only had like one dog with an intact transgene integrated in their area,
Hmm.
but the rest had just parts of that transgene integrated in places that
Yeah.
might be like concerning. But again,
Hmm.
we didn't see any pathological effects of it, but it is something that makes people worried.
Yeah. And there was like a really interesting event that they saw in two dogs
Mm.
as well, where the dogs were making more factor 8 than they thought was supposed to be
Yeah,
happening. And yeah, so they try to track down the reasoning for that. And I don't think they come to a conclusive sort of piece of evidence, but it is to do with the integrations.
yeah, that's the thing. They weren't able to catalog all the integrations, so they managed to rule out a
Hmm.
lot of different integration sites. But in the end, what they can say is that we suspect this is due to integration that we haven't detected.
Hmm. Hmm. Which again, I think that this is just a really interesting, an interesting case study in dogs when talking about gene therapies, and I'm so curious to see just more of this data. Really, like what's this data look like for the Luxturna, for the AAV in the retina? I get, retinas are a lot
Mm.
less active as well, and yeah, I'm just there's so many questions I feel like this paper raises with me in terms of how is genotoxicity really assessed, and what's the bar for getting things approved given
Yeah,
data like this? Yeah. Um,
And
yeah, it's a whole field that I don't think about that much, but is obviously something that I need to think about more. I think like everyone needs to sort of think about more.
yeah, and I think this paper has caused a lot of discussion in the field about the genotoxicity and what is kind of acceptable or not.
Yeah. So yeah, also not a surprise that this is in our top cited, and I actually think that this one will probably go on, right, in history to be cited a lot as well,
Yeah.
just because this is the type of medicine that we're really thinking about, or a lot of people are thinking about this type of medicine specifically for super rare genetic diseases, and assessing the safety is something that we just really haven't done before as a society.
Yeah, and again, 10 years like follow-up, and that's again quite an important aspect of this.
Yeah,
So,
super impressive. I'm so glad that this study exists and we're seeing the results of it.
yeah. Now, moving on to the next paper, which is Environmental Stress Destabilizes Microbial Networks. This was published in January 2021, 79 citations.
Yeah,
And
this is the beginning of, we're going to see a bunch of microbiota style papers that get a lot of attention. And everyone loves the microbiota. We think it's super important because it's everywhere, and the genes that are contained within the microbes that live on us and in our environments are responsible for very crazy metabolisms, right? That dictate the health of both their hosts, but also the environment itself. And this one is like a ecology style. Like they're looking at microbes through the lens of network parameters, saying that if they look at all the things that live in a sample, and they look at how many of them sort of cluster together in modules that they all interact in the same way. So like if one succeeds, they all succeed sort of style. So they look at modularity as one of the measurements, I guess, of robustness of the community. They want to see lots of modules, meaning there's lots of different types of groups of bacteria that are doing well. And then the other one is they're looking at positive and negative relationships, like when one goes up, one goes down, and just the number of those. And so they're going to take this analysis style and look at a bunch of sites in this particular, you had it up, yeah, figure 1. It's, what type of environment is this again?
Florida. Oh. It's at the Florida scrublands.
Florida scrublands.
Which, yeah, there are areas in Florida. I think they're somewhat endangered due to climate change. Yeah, hold on a second. I just want to check my microphone levels. But yeah.
Yeah, and so you can see this aerial image of the scrubland that they take. It's going up in elevation, and as it goes up in elevation, you get fewer veggies, fewer plants, fewer nutrients, and less water. And so it's thought that as you go up this slope, that's like increasing the stress of the environment. And so they're able to organize all their sites by the relative stress, right, based on their position in this geographical area.
Yeah, and as we look through, and I think they look at like kind of the absolute like kind of biomass of microbes, and they find that kind of that
yeah, the abundance, yeah, it correlates.
Yeah,
the abundance
as what they call like microbial stress. So they goes up, the population goes down. So they kind of define that low nutrients, low soil. That's all in very stressful conditions. And rather than they tease out a lot of like behavioral interactions in the community as well.
Yeah, you definitely have to buy into this theory of stress and like how it applies. And it's like I agree. Like I'm actually very conflicted when I think about this definition of stress on this environment. I trust the ecologists know what they're talking about, but I also think that like that stress definitely affects, like you can see it right in the vegetation, that like there's fewer things that live up there, right, as the stress goes up. And fewer things living might mean the fewer bacteria as well, right? Like it's hard to me. I guess I'm trying to understand like how much is that stress affecting the bacterial community, or is it affecting like the macroscopic life that then in turn affects the microscopic life? But I guess maybe that's the point as well.
Yeah, I think
right? Is that like all those things are wrapped up in this definition of stress.
Yeah, because when I think of stress, part of me thinks of it like who's experiencing stress in this situation? Are the microbes experiencing the stress? Are the plants?
Mm-hmm.
Or I think they're talking in terms of like the whole environment is
Is
experiencing
experiencing the stress. Yes,
that. This is
exactly. Mm-hmm.
environments with low nutrients you might get bacteria in those environments who are very adapted to those things, who you wouldn't think of those microbes as being stressed out. Like so,
Yep.
they tend to be like more slow growing. They wouldn't like they
Uh-huh.
and I think they tease that in these communities where they find out that, let's say, when you have like more nutrients around, you have more competition, you have more microbes like interacting with each other. So you have more that mix of microbes like kind of being related to each other.
Yeah.
Whereas as that stress goes up, there's less of that competition because there just isn't any energy for it, and there are
Yeah.
fewer and more slower growing microbes in the soil.
Absolutely. Yeah, absolutely. I think that's definitely how I also envision this, is that in low stress environments, there's more niches. There's more like different areas, opportunities for growth. And if one of their metrics for the I think they're called like stable versus unstable communities of microbes. Like that's the thing. The metric of modularity would be if there's more opportunities to have places to grow, then there's more niches for different communities to go, and that should correlate to the like having more modules in their network structure.
Whereas, like when you get to a stressful environment, a lot more of that,
Hmm.
but all of the energy that would be put towards competition or interacting with other is just all put into survival and strategies
Yeah.
to tolerate the stress. Um,
Yeah, there's fewer of those. There's fewer of those areas where you can actually make a living, both at the macro scale and the micro scale. Yeah.
Yeah, and they
So.
also found that like species that provide direct benefits to other community members also increase as stress increases. So thinking like cross-feeding strategies and sort of that because in interactions where microbes need other microbes in order to survive. Um,
Yeah, yeah. Actually, if we go to figure 6, I think this is like the summary, the good summary that they have of all the bar charts that they end up showing is that, yeah, so like there's more modularity in the low stress environment, and there's sort of like a equal ratio of negative and positive interactions. But in the high stress environment, as you said, there's more of the cross feeding. So it's like the organisms have to help each other out more to sustain themselves at whatever density they are in those environments.
yeah.
Um, and the final conclusion that they're trying to bring out is that the networks in the low stress environment, they are more stable, and the networks in the high stress environment, they're more unstable. But I actually don't think that I'm not really sure if that like what that definition of stability means, because in my mind, unstable means it could fall apart easily. But I'm not sure, right? Like how robust those communities are necessarily, right? Like that's something that isn't directly studied, right? There's no perturbations that are done in this particular study. It's very much like observational.
Yeah, I mean, for instance, means like prone to change and prone to like advance, and the thing that is interesting is like, I mean, I'm not sure what kind of judgments we. I think they do try to link this in with climate change because I think there's this concept of
Mm-hmm.
abiotic stress where lots of like members of like the plant community and soil community under are experiencing like environments that they're not used to due to climate change.
Mm-hmm.
Um,
Mm-hmm.
and that affects things like crop yields whereas this I'm not sure because these we're looking at organisms that are in the environment that they
Yeah.
are kind of already adapted to, so we're not seeing a changing environment. Like I remember we
Mm-hmm.
did like look at a plant like a study very similar to this that was done over the course of I think 10 years where they
Yeah,
dug up
where
the same
they
area of the soil,
exactly. Uh-huh.
and that kind of for me that would
That's a more
better illustrates.
that's more convincing evidence for like a climate change scenario, because they're actually observing the change over time. Here we're taking like snapshots, but still really useful in the way that they are able to quantify. Like to me, it's actually the paper is a lot about arguing for their metrics, or like showing how their metrics resemble something in nature, right? That like by looking at the stress gradient, you could infer these things about community structure at the network level.
Yeah, I think the network they characterize the community is the interesting part of this paper, and that can be ported over to other places because there's lots of studies out there where you take a snapshot of all the genomes and then understanding how they interact with each other. I think they've shown here can be quite an interesting and information about relationships. So, yeah.
Yep, that's all I want to say about that one. We'll have plenty more time to talk about microbiota.
Yep. Next paper is another one that is, again, quite this is quite an interesting one because I wouldn't have expected this one to be up here.
Yeah. Two randomized controlled trials of neutralizing antibodies to prevent HIV-1 acquisition.
Yeah, sounds good. Yeah, we want more antibodies to treat HIV-1 acquisition. However,
yeah, it didn't work that well.
Yeah, what they found was that the study, yeah, this is a trial. This is actually one of those prized things of negative data, where they do a test and it turns out to not turn out. Their results were they had this antibody called VRC01, which is an IgG1 broadly neutralizing antibody that was directed at the HIV type 1 envelope, and it was isolated from an HIV infected person, and they brought out, and I think they've brought it up to put it into as a monoclonal antibody treatment, which they rolled out to a couple of like kind of areas, I think in Africa and in America. It's literally right in front of me. But yeah, so they used
North America, South America, and Europe.
Yeah, they used these antibodies
and then,
as prophylaxis, and what they found was that overall it didn't actually wasn't protective on average. That as many people who were in the placebo group and in the low dose and high dose group tended to get infected. The interesting thing is when they genotype the HIV viruses, because HIV viruses, their envelope changes a lot. And so what they wanted to do is they want to classify each case based on how well the envelope reacted to them. So how well the antibody could bind to the envelope, how well it could neutralize. And what they found was, I think, quite interesting, because was when the patient had a version of the HIV virus that could be neutralized very well by the antibody, was more able to prevent the disease.
Yeah. Yeah, maybe this data is actually like a very it's almost like a like very real human data for this understanding of broadly neutralizing antibodies, because I feel the HIV research community has been thinking a lot about this particular type of antibody, where it's like it's so great because it targets a conserved site of the HIV proteins, meaning that across all HIV it's going to work. But you take that and you put it into the real world, and actually even there are still HIV strains that are resistant to these broadly neutralizing antibodies, and that impedes the efficacy of this type of intervention a lot.
Yeah, and what I feel like this paper, the reason why this paper has got cited is because there are a lot of people who are working on multiple antibody combination antibody therapies. And I feel that in every single paper that was published after this that is working on combination antibody therapy, they'd reference this as an object lesson as to why the combination antibody therapy is necessary.
Yeah, I could see that.
So,
but it's also this is something that happens like in clinical trials. We always see the data come out afterwards, because so much investment goes into setting up the clinical trial that to then say like, oh, the results weren't that great, we're not going to bother publishing it. That's not an option at that point. The type of resources that go into these types of things, like the fact that like the pre-specified endpoints and things like this, all of that, like we want to see the results. There's no way to hide, or there shouldn't be. I think it'd be very unethical to unhide the results of something like this.
I feel like there are a lot. I mean, in the Scotland
it
paper
does happen
recently with because with registered trials, I think that's changed things a lot and added a lot of pressure on people to actually publish on clinical trials. But
Right.
there has been like the reason for that is because there was a lot of problems in the past where people would just, if the clinical trial didn't show a positive thing for something, people would
Yeah.
just sit on it, and so you'd
Right.
get the effect where you get drugs that weren't very effective get come through because the one clinical trial that managed to beat the statistics and show
Yes.
that it worked went through. Um,
That's true. Maybe I'm being very optimistic, I guess, right? Saying
I
that like that things are changing, but I do think
I
that
think
it's a
things no,
trend.
things are changing, and I
Yeah.
think that we're in a situation where we can't even imagine that part, the era of the past where we could like futz around with clinical trials. Now, like there's
Uh-huh.
registration, there's regulations that go in there, um,
Hmm.
and
But not to say that there isn't like because we are transitioning from like an era where maybe the importance of that wasn't as highlighted and that it is being done through regulations, like we still have to push for that, right? Like it's something to be vigilant for, right? And to watch out. Like if studies aren't disclosing like when they pre-registered their stuff, right? Then yeah, and like that's not a good thing, and we don't want science to be done in that way. We want things to be, we want to see all the data coming out. So it's nice that this is a highly cited paper, even though the results are sort of negative. I think that that's part of like showing that, you know, the career of scientist isn't necessarily made on just positive data.
it is just as important to figure out what doesn't work. I think it's even more important to find out what doesn't
Hmm.
work than what does work, because I think there's
Hmm.
a lot more
Hmm.
in one column than the other, and so we should
Yeah.
be aware of all the stuff that doesn't work. And I'm really glad that
Yeah.
that research like this is still getting its due, that it can uh,
Yeah.
be referred to.
Absolutely. All right.
Now, the next
Let's move on.
paper, this is an
Oh my
interesting
gosh,
one.
this is so this is so different than anything
Yeah,
we really typically tackle.
interfacial engineering of bismuth 2 sulfite 3 titanium 3 carbon 3 oh, I can't remember what that last one is,
Yeah,
but
I'm not sure what T is.
I could, yeah, but
Two
um
compounds, MXene. That's the that's the chemical name for this.
MXene based on work function for rapid photoexcited bacteria killing. uh
Hmm.
so I'm going to call it Bistict MOOCs.
Sounds like a steak, BizTech.
Yeah. So this is an interesting one because this is where like semiconductor physics meets microbiology. So you can probably
Hmm.
tell we're going to talk a lot less about one aspect of this paper than the other because we're not really experts in semiconductor physics. Um,
Yeah, but in figure one, they have a real pretty image of what they grow, the semiconductor compound that they grow. And it's based on these two compounds that they have one compound, the titanium titanium stuff, and then they have another one where they grow crystals of it, the bismuth stuff. And
yeah,
so they grow the crystals of the bismuth stuff on the titanium stuff, and in that junction where between the two compounds, physics happens and electrons can flow. Energy can be exchanged.
yeah, they form a Schottky. From what I
Hmm.
brief grasp of Wikipedia showed, is like kind of like acts like a diode where current flows in one direction and not the other. So
Hmm.
it kind of
Hmm.
creates this steady state where there is like a almost like a, oh, no, like a certain
It's
line to,
Yeah, there's a polarity, a directionality.
yeah, and when like photons come into the equation, that kind of I mean, uh, enables a reaction to happen that allows it to produce reactive or oxygen species.
Yeah, absolutely. I think one side absorbs the energy from the photon, and that allows for the transfer of energy to the other side. And in that transfer of energy, this reactive oxygen species occurs, which we do know about because reactive oxygen species are also what like our neutrophils use to detoxify cells. And there's a whole bunch of things. I mean, we also don't want reactive oxygen species for our own cells because that just breaks apart DNA. So
Yeah,
this is like a real basic way of killing stuff is making reactive oxygen species. But I guess the novelty of this paper is that it's kind of a new way to make these particular reactive oxygen species.
yeah, it's
Hmm.
I mean, Yeah, because I mean, reactive oxygen species, bacteria tend to be more vulnerable to them than humans because we can produce them through our mitochondria and other sorts of things.
Hmm.
But also they're kind of bad for humans because again, they attack reactive oxygen species attack DNA, like cause aging. So
Yep.
there's uh and so like the question here is like, is this because with most antibiotics, you kind of want to have something that isn't poisonous to humans but is poisonous to the bacteria, or like there is,
Hmm.
or the degree to which it's poisonous to humans is so much lower than it is to the bacteria that it's not too much of an issue.
Hmm.
Um, but yeah, they use this brand new substance as so they do a lot of tests onto it on the
Physics that,
photocatalytic ability of it.
yeah, my eyes
So
kind of glaze over. I actually can't explain any of this, but I
no,
will trust that they did
I
it
can't.
correctly.
Literally like energy level diagrams. It'd be 20 years since I had to do them, and I was at like a grade school level, so not able to. But what I
Well,
can tell is that there is like a gap that is kind of open in one direction, not in the other, and then when light acts on it, that changes things, and the magic happens, and reactive oxygen species are produced. But
well,
this,
no, yeah, it's almost like it's the same as fluorescence works like this as well,
oh
right?
yeah,
Where like energy goes in, and then what comes out is like a photon of like another wavelength. It just so happens that whatever comes out of this, it's like a reactive oxygen species.
yeah. I mean, I presume that these aren't producing oxygen like out of nowhere. That there is, I think they do give oxygen in the specific like sources of oxygen in the
Yep.
medium. Um,
Mm-hmm. Mm-hmm.
but it's still
Yeah, so
it's
it's
a
able to
catalyst.
transfer a catalyst, that's right.
Yeah,
It's able to transfer that energy to molecules around it, creating this reactive oxygen species, high abundance of reactive oxygen species.
I mean, if they said they had an enzyme that did this, I probably wouldn't question it because they go into
Oh
deep
no,
physics.
I'm not questioning it. I'm just like I don't
Yeah,
understand what's really going on. Like I don't like see like the proof, right? Like even in an enzyme, like I think I just know the assays you might use to show that the enzyme is doing it. And here they're just using assays that I've never used because I've never had to show that I have a material that acts, right, that creates these sorts of
mm.
things.
Yeah. I mean, if this is an interface between material science and microbiology, so you've got people who are interested in new
Hmm.
antibiotics and people interested in like kind of the creation of new materials that are an engineering of them and potential applications. And
Totally.
I think this also indicates that there is, I think, because we've talked about various like nanomolecules as antibiotics. Like I think there were hairy balls that we
Yep,
thought we're talking about.
that's
But
right.
so there is this kind of ongoing like kind of study into using these nano like kind of molecules and kind of machines to as new antibiotics. And
Absolutely. And material science has a lot to bring to microbiology as well, because there's all these weird materials that microbes make, and like understanding that from a materials perspective is really useful, right? To figuring out where they can be used and so forth.
yeah,
So yeah, we definitely the merging of these fields is great. But yeah, it also yeah, we're just like not always equipped to talk about the data, especially in a sort of overview like this.
yeah,
But figure five,
figure
that's where they
five.
that's where they test it out. So yeah, actually a very simple animal model here, a type of wound
Mm.
model where the the mice are wounded and then bacteria are put into the wound, and we're seeing how quickly those things heal. And with the addition, oh, sorry, you want to say
Oh
something?
no, no, I accidentally hit my microphone, but
Oh, I thought that was like the wait, stop hands. It was the.
but yeah, I mean, I do have stuff to say now that I've screwed you up. I think
No, it's fine. Go for it.
now to put you off your game, I'm going to talk about my stuff. But yeah, this is an interesting thing because it's a soup where they they cut an animal, they add in the bacteria, and that causes an
Hmm.
infection, and then they add their kind of their product and expose it to near infrared radiation for about I think 10 minutes, and that activates the photocatalyst, and it causes it to kill off the bacteria, and they don't really measure the yeah, they don't really measure the death of the bacteria. What they measure is the rate of wound healing afterwards.
Hmm.
Um, and again, we don't, and they do like try and like look at kind of whether this product is toxic to internal organs. There are multiple shortcomings of this model that I don't think this is going to be a replacement for an antibody. I think this is very much a proof of concept for the sake of a proof of concept. I don't think it's
Yeah. I almost wonder why animals were used in the first place. Like, they could have done this with just some bacteria probably and have interesting stuff.
I think they were interested in toxicity, but I'm the way they kind of dropped it on the surface, so
Mm-hmm.
that's probably not going to get in too deep. So I
Yep.
don't think this would be. Firstly, the photodynamic therapy, there is some limitations to it being used in deeper tissues.
Mm-hmm.
For instance, light can only penetrate so far through tissues. I mean, notice here how I'm not see-through. Light, fine,
But that being said, surface injuries are also like a really big category of injury, and so like just being able to deal with things on the surface may be quite useful.
yeah. I think like things for like wound dressings and plasters, I think that's where this kind of thing will be quite useful for, and that's what
Hmm.
they. I think that's the function they're looking at it for.
Yeah, I'm actually more like what when I was thinking about this material, I was more thinking like, but what about like just places that you want to be clean from bacteria? And maybe
Yeah,
this is like interesting like anti-fouling stuff or like I don't know the
yeah,
stability of this particular catalyst, but maybe they can just like put it in some porous structure and like coat something in that structure, and then anything that grows in the structure you just irradiate with this thing and everything dies inside.
yeah.
Like it seems kind of there could be those types of applications as well. Doesn't have to be biomedical.
Yeah, I think that's like coatings again. It's a really big science. Biofouling is
Hmm.
really like a real problem
Hmm.
for a lot of places. So again, this is something that could be quite useful for that. Um,
Hmm.
but yeah,
But I
lots of
think there's excitement over biomedical applications. So often we'll see as the roundup figure for something like this, some sort of biomedical application.
yeah. I think that's
Yeah.
absolutely correct. Yeah,
And why this is high. This is really high on the list in terms of the number of citations it got.
it is. It got so at this point this we've let's see. Let me double check this. Yeah, it's still at 84 citations. We haven't broken the 100 citation mark yet, but it's still.
Hmm.
I think this is going to be the next one is good. We're talking about will break the 100 citation mark, but this is like yeah, very high because I think that this is a hot field at the moment.
Mm.
The idea of engineering materials and antimicrobials, it, it's there's a lot of like interplay there that's going on at
Yeah.
the moment. I think this
But
the,
maybe also some of the physics that we weren't sort of capable of judging, like we just don't understand the significance of like how great that this material is. Like maybe
yeah.
outside of the application on biology, there's something really interesting about the material they were able to make and characterize.
Yeah, so it could be that because this is a paper that is hugging two disciplines, it could be
Hmm.
really a big thing for the engineering material side of it, and
Right.
we're just not seeing that.
Right. Like, because I'm thinking like this is also why I thought the HIV paper, the capsid one was so interesting because it's like there's also a big like cellular implication to that. So like all of cellular biology is also following that literature. Yeah.
Yeah,
Okay. But moving on, now we're breaking the 100 mark.
yep. The 100. So this has got 101 citations, and it's
Hmm.
titled "A Genomic Catalogue of Earth's Microbiomes." So they're aiming big. This is a big one.
But also not much more to explain there. Like
No,
this
no.
this is
Yeah,
exact.
exactly that. Um,
They do talk about this concept that I hadn't really thought about before, MAGs, metagenome assembled genomes.
mm,
So with basically the scale of data that we're able to accumulate these days, they're just like just sequencing from environments. So whatever environment they find, get all the sequences back. You get tons of these fragments, and using the magic of computers and algorithms, being able to match up the overlaps, or even having like rough, yeah, I think it's overlaps that they're using to make big contigs that then can get bigger and bigger, right? Small things match up into bigger things until you get something that sort of resembles a genome. And that's the technology in which they're extracting genome level information from essentially just like bulk environmental reads.
yeah, it's putting together a lot of like data that was previously unpublished along with published data to get
Hmm.
an idea of micro metagenomes across the world.
Yep.
So yeah,
And
so the GEM catalog, the Genomic Catalog of Earth's microbiomes,
mhm.
and
It can
a
essentially.
lot of that.
Oh, sorry. Go ahead.
Sorry, I was going to say that a lot of it is like this is a resource that people are going to be using in the future. The thrust of this is kind of like showing that we have this resource, we're going to hand it over to you, and here's a few examples of what it might do.
Absolutely. And just knowing how much microbiome research is happening, this is going to be cited a lot because it's like, oh, we found it in this database, right? Or we're following up on this unknown thing in this particular database. I want to draw people's attention to figure 3 because like I talk about when I think about microbes, I always think about like how great it is because there's so much diversity. There's so many different things that we know, and these types of catalogs sort of continuously push the sort of our understanding of how big the world is in terms of what lives and how much we don't know about what's living. So on this little crazy tree of life, semicircle, everything in green is what they've added to the existing tree. So they essentially like doubled, I would say, like our understanding of like what's out there. And you can see it's like pretty well distributed amongst all these different trees of life. Even the eukarya, right? They're finding all these new eukaryotic sort of things going on. Yeah, I don't know. That's what's here. They do point out a lot of different things, just like how much the limits of the things that we know. Like I think they looked at the saturation. Let's see, where's one of these saturation curves? I feel like there's something here, or maybe that was the other one.
But yeah, I think they look at the biosynthetic gene cluster. So like how people who are bioprospecting would want to see like, oh, what's the capacity of microbiomes to produce like new products, and they kind of hone in on that and look, they find that there's lots of these new like kind of things that we know these genes are related to creating new like kind of products, but we don't know what those are. But this kind of like gives a kind of a little taste of thing. Oh, there's lots of that. They also I think they find a bunch of new virulence factors as well.
Yeah.
And also like thousands of new virus host interactions as well.
Phages,
yeah, lots of phages, and so they kind of like show that there's lots of like data you can get out of that. If you're looking for like the next new antibiotic or next new kind of treatment, then this is like another database that you can look into to
Right. Yeah, actually, I think I was thinking about another. It must be an upcoming one where they also look at eukarya. This is just bacteria and archaea.
Yeah.
Yeah. So there's also like all these single-celled eukarya that like might be able to be assembled, but they haven't put it into this diagram yet.
But yeah, I think that would bring us to the next paper, which is another microbiome paper. 149 citations, microbiome connections with the host metabolism and habitual diet from 1,098 deeply phenotyped individuals. So a couple of big things here. Firstly, diet. That's a big topic for nutrition. Microbiomes, another big topic. And I think I got 2. Unfortunately, I didn't. But yeah,
yeah, those are big. It's hard to take it's hard to associate things with diet because we're all eating whatever we want typically, and like to study this, you'd have to give people a lot of questionnaires as well as do follow-ups where you're not just relying on those questionnaires. So they also have clinical data here where they're sampling blood at certain points and looking at all these different markers. It's not an easy data set to collect. And so I think that that's probably what drives the popularity of this particular paper, that it is just highlighting some very difficult to collect paper that people are obsessed about, right? Like microbiome and health, especially as it impacts on diet or as diet is impacted. That's really big. And this paper has the reading of something that is also trying to speak towards policy a lot. Like they use definitions in their diet data. They subset foods and things by groups that I guess are accepted within policy circles, right? Healthy foods, right? It's like some broad overview, and they're seeking to make some recommendations, I think, from this data set.
Yeah. So let me see if I can pull out what things stood out for you. So I mean, I think it's interesting the way they did this study. So they first started off with a population of twins, then that further to other people, and they had a validation cohort, which I always think is very important for studies like this, where if you, in your first cohort you're kind of fishing for a hypothesis, and then your validation one you're looking to see whether that kind of relationship still holds up.
Yeah, well, they're doing such large comparisons, right? They have a huge set of microbiome data that's like, so many different variables. Then they have this huge set of like the diet characteristics and the levels of different different markers in the blood. And so they're using, of course, machine learning and training, right, on that data to find trends, and then taking that model and seeing, well, now with the data that wasn't in that model, but essentially should be the same, do these trends still hold up? So they often say like the model's trained on the UK cohort, but it is validated in the US cohort, that these trends are similar, right, between the two.
Yeah, and I think I mean, the interesting thing, well, there's lots of interesting things there, but one thing's, I mean, firstly, there's a lot of poop sampling in this. A lot of this is taking like microbiome, and because I think with a lot of microbiology, like microbiology data related to diet, people often think of like, oh, well, let's find what help people's healthy gut microbiome is, and then transfer it to someone who's unhealthy and see whether that makes a difference. But I think this is more using the microbiome as a biomarker to
Mm-hmm.
indicate potential diseases. So
Mm-hmm.
I don't know if you've heard of like the digital toilet concept, where like your doctor, so if you poop, your toilet
Yes.
will take a sample of that, run it through a bunch of diagnostics, and then tell you whether you're sick or not. I think this kind of relates to that, where they're
Yeah.
talking about like, okay, we're analyzing people's poops. What can we tell about? And so from this, they can tell people, they find associations between diet, and they kind of make that judgment on what healthy and unhealthy like plant and animal based foods.
Mm-hmm. Yeah, and they find specific bacterial clades that are good markers for these things, right? So they have all this clinical data collected, and they can say, oh, if you have more of this bacteria, then there's a high likelihood that you'll have a higher blood serum level, right, of this particular thing. And like those associations, again, it's this biomarker type thing, and maybe useful for making diagnostic tests in the future, or understanding, or just interpreting people's gut microbiome.
Yeah, I mean, they find things like the gut microbiome can predict insulin concentrations better
Mm-hmm.
than, say, glucose levels, or give you an idea of like how your metabolism is doing. Um, and they also do.
They also have
Sorry.
hilarious soundbitey things like coffee has the strongest effect on your gut microbiome.
I mean, it is a diuretic, so it probably would have a major effect. Uh,
Yeah.
but yeah, I mean, lots of like things that I feel that I am concerned might get misinterpreted, but I think
Uh-huh.
the researchers here have like a very clear idea of what they want to show,
Yeah.
and I think it's going to be a useful resource for people to refer to back to when they're making their own study of the gut microbiome.
Yeah, I think that's definitely why it's getting cited a lot, though, right? Because of those one-off tiny observations that can get picked out here, can then go into the introductions of so many
Yes.
other papers that are actually trying to study these health things and trying to, prop themselves up with a little sliver of microbiome data to support their hypotheses.
Yeah. Yeah. I mean that's what I think. But then yeah, that's probably the most cynical interpretation, but it might be true.
Sorry, maybe I'm very cynical, especially when it comes to like these very hot topic areas,
Yeah.
right? Microbiome and diet. Like I can't like, yeah.
Yeah. I mean they find like metabolic markers of obesity and other sorts of things, and I'd be interested to see where this goes.
Hmm.
Because I mean, how much more subtle can you get with it, and other sorts of like it's an interesting new way to study biology by looking at the microbiome as it relates to human health. I think this is very like in it's gonna be interesting to see how this work turns out.
Right. Oh, but this is the one where they do have eukaryotic sequences
Yes,
in this particular mix. And so I did want to point out that they did talk about
yes, which we
blastocystis,
covered
which
last week.
which we covered last week.
That the blastocyst is a marker of improved postprandial glucose responses. So after you eat, but having lots of blastocysts indicates that you have good control of your blood glucose. So that's good, I guess. I don't know what that relationship is or how it acts, but I think it's just we're gonna have to do more research as needed. Um,
Yeah, Andrea added some other microbiome trivia inside of our chat. And so, yeah, like people are always finding interesting associations, I think, with microbiome. And it's really like sort of taking that information and then trying to, well, at least from our perspective as microbiologists, right? Taking that information and following up with some interesting mechanistic conclusions. You know, I'm also really happy if it somehow gets all the way to like a real clinical study of some sort. But I think the first steps really coming from these large surveys, in my opinion, would be, yeah, to try to tease out a mechanism of some sort and then create an animal model that you can study that in.
Yeah, and I think the next paper, kind of dreading the next paper,
Dreading the next paper, okay.
safety and
Oh,
immunogenicity of an anti-Zika virus DNA vaccine. 133 citations in 1 year. This is like a big study. Zika virus was also like a very is one of those brand new emerging infections that was discovered like in the 70s in Africa and then wasn't really paid attention to until like very recently in 2014 or 2015. I can't remember the exact date, but where it kind of went global, spreading uh from Polynesia all the way to Brazil, and very similar in the way that monkeypox was again known in Africa and has only recently become like a global thing. Same things happen with multiple diseases, so it's
mhm.
something that's of a lot of interest. And so this is a
And
test of a new anti-Zika virus DNA vaccine. um,
which we've talked about this particular vaccine delivery mechanism before,
yes,
right?
yes, we have.
It's a
This
type
is
of
my nemesis in terms of it's an OVO. So if you haven't watched the show previously, we did for a long time we were looking at chasing it after like the new SARS-CoV virus vaccine candidates, and one of the early ones was the one by this company called Inovio, where they'd have this DNA and you inject it directly into someone's arm, just normal like DNA on its own, and then they'd inject you. Then while they're injecting you, they also electrocute you cool through operation where you apply an electric shock to cells, little like holes open up in them that DNA can bust through, and that's a way that cells can take up DNA and thus take up a DNA vaccine. Um, and
Mhm.
yeah, this is like my least favorite of the vaccine candidates because I don't. I mean, look, I'm happy if it works. If it works, that's great. I absolutely love it. But the prospect of being injected with the vaccine is something that is not something I'd like to. If there's any other option for a vaccine, I'd love it. But if it's the only option, then yeah, sure, I'll take it. But not a fan of the cell
Yeah.
electric system.
I think we actually even looked up in our early days of like reading about this vaccine. There were studies saying how it feels to be
Oh
electrocuted
yeah,
by
on a
this.
scale
Yeah.
of 1 to 10, where 10 is the worst pain you could ever imagine feeling, and 0, it gave. So, meh.
But yeah, again, this is one of those, it's a vaccine type that ostensibly would be very easy to make, right? Very easy to make variants of, because again, it's just producing nucleic acid, which we can vary much more easy than varying protein. And yeah, so in that way, it does have like a lot of promise. And yeah, in this study, it's actually not a very big study.
No,
It's
it's 20 people.
Yeah,
And
40 study participants enrolled at 3 clinical research sites.
yeah, it's not so again, like none of the like complex lipids or like nanovesicles. It's just and they do inject a lot of DNA. So for instance, for the mRNA vaccine, you got 60 micrograms or in the microgram range, whereas here you've got people being injected with about 2 milligrams of DNA, which is quite a lot. But again, the important part of it is that they do find that it kind of works. Um,
Yeah,
that
well, they're using the serum right from these vaccinated individuals and putting that serum through many different tests. So like the idea is that it's like a lot of looking at the antibody correlates, right? So does the serum have neutralizing antibodies against Zika? Can it protect mice from Zika? And then they also, because they're taking blood, they can also look at some markers of cellular immunity as well.
Yeah, so I guess I need to qualify my it works. It works because it's a phase 1 study, so they don't actually test whether people like it's not they're not doing it to people get infected with Zika. What they test are those important proxies of immune effectiveness. So does it produce an antibody response? Does it produce a cellular response? And yeah, they do this weird thing where they take some antiserum from the people who are given this vaccine, and they test whether they can stop mice from getting. So this is effectively like trying to, almost like a neutralizing assay where they're trying
like
to
with a more complex outcome, right? Like it has like something, oh, like these mice would otherwise die, so the neutralizing ability is able to save mice. Maybe speaks a little bit to the mechanism, right, of immunity, but we don't know what that is in humans necessarily, right?
Yeah. And what they show is that they produce antibodies, and these antibodies can like hit the virus and stop it from working. So that's an important first step.
And yeah, so why is this one cited? Why is this so highly cited? Maybe partially because there are SARS-CoV-2 therapies that are based on this technology, and this is like as seen in a phase 1 study. But also because I think like Zika is right thought of as like an emerging pathogen with no current therapeutic options, and so having this, like, there could also be excitement there, right? To have this technology move through the clinical pipeline.
Oh, I think I know what happened here. So in 2017, and what we see sometimes, what you see with a lot of like big publications is that there's a preprint that let's say you produce a preprint of your article and then you send it off to a review. The longer that review takes, the more citation that preprint will take. So when the article is eventually published, those citations will move over to your produced article. So
so this could also be, these could be 2017 accumulating since 2017 citations.
Yeah, that is potentially what happened here. I mean, it's not something that is particularly ethical for journals to do, but sometimes just happens by accident.
So it could be that. Do you think my hypothesis could also be a possibility?
I think
just having
I
knocked on.
I believe that your hypothesis is that because firstly, Inovio is like it is a new vaccine technology, and it is very firstly, you don't need it's very tailorable. I think as well because you can do any DNA with it, whereas I think with the kind of what's it, liposomes, you need to tailor those to the specific structure. You need to do all sorts of alterations. With this one, you can create something very quickly, and I think that could be explanation. I think also Zika virus is a really important pathogen.
But yeah, that's that's what we got. Phase 1 clinical trial, second most cited paper in 2021, as long as they didn't carry over their citations from 2017.
Yeah, I mean, because it was published in September 16, 2021. So like imagine those citations times like because a lot of the other things like we've seen up here have been from January. Right. So these would have been collated over a very short amount of time. So maybe there has been an explosion in Zika virus research.
Yeah, actually, I actually think yours is more parsimonious now. Your explanation is more parsimonious.
I I know. Apparently cynicism is winning today.
But all right, let's go to the top most cited paper of 2021 that was not a COVID paper.
A unified catalog of 204,938 reference genomes from the human gut microbiome.
Another catalog.
Yep.
Not to say that again, microbiology, I love it. I love that there's so much excitement over cataloging everything that's living out there. But yeah, like it's just these are definitely resource style papers that have done an amazing amount of work to bring together a database that other people can look in. But in of themselves, the looking at these databases doesn't. I guess to me it just isn't as exciting in terms of what it's telling us. But this one is the one that has the rarefaction curves. So I do want, we can talk a little. There are some things I want to highlight here.
Yeah, another page. Yeah, metagome. So mags, I think, are so hot right.
Yep.
The thing that,
apparently.
Yeah.
Apparently, yeah.
But yeah, they gathered all these isolate genomes and from the human gut microbiome. So this is different from the all Earth genome, but we do get microbiome things from all over the world.
Oh, can we pause? Can we pause for a second? Do you know if there's a phase 1 COVID vaccine with Inovio, or was it's still preclinical?
Oh, I think there was a phase 1 study on it, so it did make it to phase 1. It just was.
Gotcha.
It made it to phase 1 too late because I think Inovio had some production issues and there was a legal problem that caused some delays in their. But I know that they did get to phase 1 at least.
Right. I think we might have even talked about the phase 1 trial then, if it did get there, in the Vaccine Olympics. One of the many Vaccine Olympics. We might be able to point to that. But it's also not as organized back then. So we understand if you're not going to dig through our backlog to find that particular video.
I think it was one of the first, because I think it was one of the first papers we did a deep dive on, because I do remember trying to do like an animation showing what electroporation does. But yeah, I definitely recall like having to go through press releases on like the subject of whether Inovio will be, because again, at that time we're looking at the race, seeing like, okay, which one's going to go first, and I was just going, please, anybody other than Inovio, please.
You weren't looking forward to being electroporated.
Yeah, they do great work. I know. I mean, it is definitely a good vaccine candidate, but my personal preference would be don't electrocute me.
We did cover something in May of 2020, May 17th of 2020. A DNA vaccine induces SARS coronavirus protective immunity in mice. I don't know if I don't know if we looked at the phase 1, but we did look at the at least the early one.
Yeah. I am aware that a phase 1 thing does exist out there somewhere.
Yeah, and there's definitely phase 1 for MERS, right? They were also trying this. They were also trying this for other viruses. Okay. Anyways, I just wanted to get to that the question that was asked.
Of course. So
mags are hot. Mags are hot right now.
Yeah, mags are hot right now. I mean, so a lot of the gut microbial species they found lack actual isolate genomes. So I think that again, when you do these studies, a lot of like unknowns appear, and it kind of pushes back the frontier and shows us kind of what is still left to find out.
Right. Yeah, and this study is specifically looking at just the gut, which is like it's a very complex and very relevant microbiome for us to understand. But like some of these genomes are going to be represented also in that global catalog, because I'm pretty sure the global catalog included human as part of their set.
Yeah, and that'll be interesting in terms of like the all the other things that they were talking about with their
like some of the overlaps I think would be very interesting. So in figure 2 though, this is what I thought was in the previous one, but is actually here. In figure 2b, they have this curve, right, where they look at of all the species they get, right, as they add in more of the genomes, how many new ones do they find? So if they remove all the the singletons, meaning the only single mags that they paired off with something else, they add these sequences and they see, oh, without the singletons, everything levels off. And if it levels off, it means that as they add more genomes, they're only getting duplicates of things, so there's nothing new to find. But when they look at the singletons that they got, that curve doesn't seem anywhere near leveling off. And so the implication for this type of analysis is that there's still more to find in the gut microbiome. That even with this most recent catalog of genomes, we still don't know everything that lives within the gut. We don't have the genomes of everything that lives in the gut microbiome.
Yeah, that is like an amazing thing, because I think the singletons are like, you see them in one kind of data slice and you don't know whether it's actually representing something real or not, I guess. And because people eat all sorts of stuff, so I mean, stuff in the environment, there are all sorts of microbes in the environment that can end up in the gut and end up here, so we don't know whether...
transiently going through the gut. Absolutely. So that could be the explanation as well, right? That we're just being constantly peppered. But some of those things might end up having a hold. I think they're not using just 16S here for B. Like, they're looking at it from genomes. So like, there has to be enough of the bacteria in the samples to be able to get all the fragments to say that something passes their quality check for what counts as a genome. So like, it could come transiently from things we eat, but like, yeah, they are somewhat established in our gut.
Yeah, I think that, and yeah, the depth of and again, it represents like countries in the US, China, and other places more, but there are lots of places on this map that are in gray or in like blue that aren't very well represented. So it's easy to to think about potential situations where in certain localities you're going to get people with a certain microbiota that wouldn't appear in this data set.
Absolutely. Yeah. I mean, yeah, like that curve isn't, yeah. So in this data set, we're not even capping the full diversity of what's there, let alone the global data set.
Yeah, like, it doesn't represent people who like eat Ethiopian cuisine, or like you've got a lot of people who eat burgers, I think, showing up in here, but not necessarily as many people who eat borscht or other sorts of like, there's a lot of diversity in microbiome, in like food and microbiomes out there that might not be represented in this data set. Not to
we saw that in the diet study, right? That certain foods create very strong signals. Maybe there are foods that are creating strong signals that we're not seeing here. Anyways, it's another catalog. You can say some of the same things about what's going on. And sorry, not to diminish it. I'm sorry.
I don't want to say we're not dumping on this this fantastic study where they take tons and tons of data, and the first thing we go is like, well, if I want to well, where's the sushi in this one?
I think it's like,
this is actually,
maybe I'm disappointed because I can't dig into this maybe as deeply. Like, I feel like a lot of reading this is reading like summary data, right? So like, it's like understanding like the overall shape of what's going on here. And I'm really interested in seeing where people take those ideas and where people drill deeper in them. And maybe it's just not as satisfying to me reading this because I'm not so entrenched within these methods. It does come back to that, right? My inability to interpret the methods as deeply is like part of why like I glaze over some of these things.
Yeah, it's not quite as like fun to read as like last year we had the AI generated antibiotics, which was really like had a lot of depth to it. Whereas this one is like, it's almost too much.
Well, this does have the depth. It's just like, I'm not. Yeah. It doesn't really strike my interest level. Yeah.
That paper last year was like a gentle sip. This is like a fire hose of just, there's so much that it's hard to kind of get one story.
Yeah. But in terms of trends, right, like this really speaks to the trends of top cited papers in the last year, that microbiome research is something that is within the scientific eye right now. People are I think people have access to this data in a scale that is sort of unprecedented. And it's probably happening in a lot of different fields as well, right? Like not just microbiology, but cellular biology, cancer biology. People are really tapping into the genomics and the big data genomics of it all in order to try to find answers. But at the end of the day, I'm an experimental microbiologist, and I'm curious to know what this means for folks at the bench
Yeah.
and what sort of phenomena could be uncovered with some of the leads here.
Yeah, I think that brings us to the end. I mean, we have some brilliant comments about microbiome, because I think about microbiome, and I think some interesting things to look into for the future.
Mm-hmm.
I mean, again, it's interesting because the comment was about mitochondrial and oxygen availability, which is something that, the gut has lots of different environments within it. There's anoxic, there's oxic. It depends on like the thickness of like the mucus layer. There's a lot of like interesting things going on there, because and I think that's why people have this interest in it, and why a resource like the unified catalog of 204,938 reference genomes will be something that's interesting for people to look at in the future.
It's a very impressive number. I don't think there's many things in my life that I count to that level.
Yeah, I would have rounded up to like 20,000, 200,000 would be impressive enough. But
I'm cowed by the.
If only they'd put 204,938 and a half, then I'd be more impressed. But
is there anything else you want to say, sort of like about the trends that we saw over this top 10?
Well,
or
I think the trend we're seeing is that firstly, mags are so hot right now.
Yeah, microbiome stuff. There's actually
a lot of interesting. I mean,
a lot of their clinical studies, right? That actually like high-profile clinical studies make their way into this list. Maybe we saw something about interdisciplinarity, right? Like I was thinking the material science interface with the reactive oxygen species, the cellular science interface with the capsids, maybe even some like strange like neuroscience. I think neuroscience interface with the seeing stuff, the gene therapy.
I think that gene therapy again. Like we had 2 papers in the top 10 that were very high up for gene therapy, and I think I'd even count the Inovio paper as a form of gene therapy as well.
Yeah, absolutely. I mean, you could imagine that platform being developed for therapeutics for certain gene diseases.
I mean, this is part of why I'm thinking like gene therapy is like kind of having its day, because nearly everybody, quite a lot of people got like the Chadox vaccine, which is an adenovirus gene therapy. So that feeds into that kind of idea for me.
It does, but I also I do think that there's a lot of space, and maybe we should be talking about it more, right? Like what goes into thinking about whether these technologies are good or bad, and like more than just because it sounds very sci-fi, but it is happening right now, and so it's good to know like what the concerns are, what are regulatory bodies looking for, right? Like this 10-year follow-up. Right, I'd be like, wow, like that's something really interesting to know that could be done, right? Could be a gold standard that is set, right, for certain things that come out. But then, balanced against, the cynical part, balanced against like the need to get drugs into market, and of course the needs of patients as well, like who want those drugs.
Yeah, and I think that kind of draws it to a close. I think the interdisciplinary aspects are very important. The resource aspect, so creating something that other people can use, is also something that is very good for the community. We haven't really been giving them as much credit as I feel we should. But it's very hard because they're not fun stories. They're not like they're very much like here are my Lego set pieces. You can go play with them.
Right.
And from our perspective, we're like, oh, this is just a box of Legos. But from someone else's perspective, like that's a pirate ship. That's the Millennium Falcon. That's what we're gonna.
No, I'm glad that you. Yeah, please let's. I do. I want to lift up those papers because they are really important. And actually, that reminds me. Also, we saw a negative data paper. It was a clinical trial.
Yes.
Right. But like negative data is also really important, and I'm happy that something made its way into the list from that perspective as well.
Yeah. So thank you very much for joining me for this. This is a longer show than usual.
Yeah, thanks for everyone who's stuck around, and if not, you're just catching up over the asynchronous. We're happy that you're watching this extra half hour of content, 40 minutes of content. Next week we won't be. We're going to try to switch to every other week. So we won't see you guys next week, but we'll see folks the week after that. I'll make sure to send out some reminders on our Mastodon and our Twitter.
Yeah, we want to be very enthusiastic about microbiology and somewhat qualified. It's possible we didn't get everything right. Science is about thinking critically and asking the right questions. And if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter, as I mentioned, with @MicroTWJC, or on Mastodon, where we're MicrobioJC. We both believe that peer review is a process that we can all participate in, and so we hope that you've had a good time listening to us ramble on about microbiology today. And if you have something to add or found something interesting or found something unclear, just let us know in the comments.
Yeah, it has been a pleasure, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. Goodbye.
Bye-bye.